Hey dear students, welcome to my YouTube channel. So in this video we will study about the fasciola. So this fasciola word is come from the Greek word. Okay, so fasciola means small band, and where is hepatica word come, that means hepatic word is related to liver. Okay, so there are two species of fasciola. First we have Fasciola hepatica, which is also known as the sheep liver fluke. Liver because of hepatica. Why it is called the sheep liver fluke? Because it was first time described in sheep as a liver rot in 1379. Okay, and second species we have the Fasciola gigantica, as the name suggests, giant, large size. Okay, so Fasciola gigantica is also known as giant liver fluke. So this is a species of the Fasciola genus, and they are not only found in the humans, it also found in the veterinary, like animals, sheep, goats, camels, cattle. These all are the domestic animals. Okay, next we have the epidemiology. So now if we talk about the first, that is the Fasciola hepatica. So it shows the worldwide distribution, that is the cosmopolitan type of distribution, and it is the common genesis throughout the Russia and parts of South America and Africa, and most common areas are the between it commonly occur in the like livestock animals like sheep, goats, camels and cattle, and humans and snails. Okay, so here snail is the intermediate host. We will study in the life cycle. So now if we talk about the Fasciola gigantica, so it is endemic in the Africa and Asia. Next we have the morphology. Now if we talk about its egg and adult, so adult worm is like a leaf shaped and approximate 30 to 12 micrometer. Fasciola hepatica and gigantica is approximate 25 to 75, okay, into 12 micrometer in size. Gigantica is largest in size as comparison to Fasciola hepatica. Gigant, as the name represent giant size, it is the 30, and this is the approximate at least 75. Okay, so there are a distinction between these two species. If we talk about the Fasciola, there is a presence of shoulder. Okay, and there is a cephalicon, that is the anterior prominence. Next we have the oral sucker. So it is situated in the cephalicon, and they are equally sized ventral sucker. They both are the equal sized. Okay, so the adults are the monoecious. Now if we talk about eggs, so the eggs are large and yellowish brown in color, while stain void, and measure approximate 130 to 150 into 60 micrometer in size. Okay, so next we have this further stages like larva, which develop into the sporocyst, stadia, and finally into the metacersaria. We will study it, the development stages in the life cycle. Next we have life cycle. So we will do the life cycle from the diagram. So we have three phases. First we have migratory phase, where it was like the metacercia, that is the ingested from the aquatic plant, whether it is consumed by humans, cattle or goat. We are consumed here metacersaria by the aquatic plants. Okay, so human consume this. Okay, so first the metacersaria ingested by the host duodenum, that is the small intestine part. Okay, so it penetrate into the intestinal wall to reach the peritoneum. Then they further burrow into the glycan capsules and enter the liver tissue, and finally infiltrate the bile ducts. Okay, so bile ducts involves the 6 to 7 weeks. So this migrate like first it comes to the intestinal, then glycan capsules and liver tissues and bile ducts. So this is the migratory phase, because this migrate from one organ to another organs. Okay, so next we have the sexual phase. So the metacersaria settle into the adult fluke in the bile ducts. Okay, we can say bile ducts, gallbladders and liver tissues of the mammalian host. We are considering here the humans. Okay, so after the maturation they start producing eggs. Okay, so they have a long lifespan of more than 10 years. So the unembryonated eggs, you can see the unembryonated eggs released into the environment. How they penetrate into the environment? By the waste material, that is the feces. Okay, so about 3-4 months after the infection. Next we have the asexual phase. So this comes from the feces, that is the from the waste material. Next is the asexual phase. This is the sexual phase and this is the migratory phase. Third we have the asexual phase. So in fresh water, body of the this is the unembryonated eggs. Okay, so this is the diagnostic stage. So it is developed in the this miracidium egg. Okay, so then this egg transferred to the intermediate host. So here intermediate host is the snail. Okay, so this is the snail species, which is the species specific snail. So in the snail, this miracidium goes through the sporocyst stages, and after the two-third generation of the Fredia finally produces the, here is the Cerceria. Okay, so this is the Cerceria, and this Cerceria are released into the out of the snail after the 4-7 weeks and get ancested into the aquatic plants. Okay, so they remain in such a state until they are ingested by a new definitive host, or we can say the primary host. Okay, so this life cycle is continuous like this. So first we have the migratory phase. Okay, where the human consume this, and this Marsaria which are consumed by the aquatic plants are transformed, are migrate from the organs to organs, and lastly they come into the liver tissues or bile ducts, and then there is the uh, another maturations, and there is a diagnostic stage, that is the unembryonated eggs are released by the waste materials, and then the asexual phase, that is the miracidium, which are uh, in the intermediate host, that is snail, and then develop the further stages Cerceria and the metacerceria. Okay, so this metacerceria further consumed by another host, like we can say cattle, sheep, goat, and the human. So here snail is the intermediate host and human is the definitive host. Okay, next we have the pathogenesis. So this disease cause the tissue destruction and inflammation associated with the larval migration through the intestine, liver parenchyma, and it also causes the allergic reaction to the parasitic antigens. So there are some features which are seen due to the fasciola, like the bumps reside in the bile ducts and stimulate the chronic inflammation and the fibrosis. Okay, so inflammation and fibrosis. Next is the, there is a granuloma formation around the eggs and bumps which develop into the abscess. This is followed by the fibrosis. So it mainly causes the fibrosis. Next is the clinical features. In clinical features we will see the acute and the chronic infection. In acute we can see the symptoms like the nausea, diarrhea, bloatings, cough, etc. Okay, but in case of chronic infections, so the adult flukes can live in the humans up to 15 years without any symptoms. Okay, so but after the chronic infections after 15 years, it can cause the inflammation, obstruction of the large bile ducts, etc. It also causes the hemorrhage, that is the rupture of blood vessels, so etc. Next we have how we can diagnose it. So we can diagnose it various techniques like microscopy, histopathology, serology, and other methods. So first we have the microscopy. So while the bed mount preparations are perfectly concentration techniques, it can use to diagnose by the microscopy technique. So by visualizing the eggs or adult worms, we can use the direct microscopy of stool, duodenal, aspirate, or bile, and it is also used for the ERCP. Okay, next we have the histopathology. Histo means tissue. So parasite fragments and eggs can sometimes be seen in the surgical tissues or the biopsies. Next we have serology. So the commercial available serological tests are the like we have secretory antigens which helps in the early diagnosis before the eggs detected in stool. Next we have the copro antigen detection. In this ELISA based tests are used. We have the treatments which are used to treat the fasciola disorders like triclopentazole and nitrogenamide, etc., methanol. So these are some drugs which are used to treat the fasciola disorders. How we can prevent the transmission of fascicular disorders? So we should stop the raw or undercooked fish or other aquatic plants consumption. Next, there is a proper sewage system, underground system of the waste materials, and there are some drugs we can use it. So these are the prevention and control of Fasciola hepatica and Fasciola gigantica. If you have any query, you can simply comment in comment section. So that's all about today's video. I hope this video will helpful for you. Thank you.